Now, there's another phenomenon related to co-translational translocation, protein sorting, that we have to deal with, and it's related to what we were just talking about with respect to the Brownian ratchet mechanism. So we can start from there and elaborate a little bit on what happens with the nascent chain with respect to the problem of protein folding in the lumen of the ER specifically. So there are, in fact, many processes that act on the nascent chains in the lumen of the ER that are specific to that particular environment. For one thing, it's often the case that secreted in integral memory proteins that face the outside of the cell are glycosylated. They pick up oligosaccharide trees, basically, that are attached to the polypeptide chain. That actually occurs very early. It would be going on right about now in this diagram in the overall process. And also there is the distinction in the environments of the lumen versus the cytoplasm that I've hit pretty heavily, so I don't want to go into that any more than I have to, but those environments are really quite different chemically, and among the differences are the fact that the cytoplasm is much more reducing than the lumen is. So we think in terms of the lumen as being an oxidizing environment, as we do the extracellular aqueous phase. So the significance of that is that disulfide bonds form in the lumen and not in the cytoplasm. The reducing environment in the cytoplasm promotes sulfhydryls, whereas the oxidizing environment in the lumen and extracellular space promote disulfide. So those are going to be actively formed about this same time as well. Both of those things are significant in that they interact and essentially cooperate with BIP in the assisted folding of the nascent chains in the lumen. And it so happens that those same phenomenon, the glycosylation and the disulfide bond formation, are also very important in solving the problem of misfolded proteins in the lumen of the ER. Now, the sad fact is that misfolding occurs in the lumen of the ER for many of the same reasons that occurs in the cytoplasm. There is an additional need to handle proteins in the lumen of the ER in such a way as to get rid of them, even if they're folded correctly. And that has to do with situations in which there is, say, a multi-subunit complex, and the co-translational translocation process leads to the synthesis of too many of one type of subunit. So there's essentially a leftover spare part that needs to be gotten rid of. So that is handled in exactly the same way that misfolded proteins are handled in the lumen. And both of those are related to the fact that those things have got to be sent to the proteasome. But the proteasome is in the cytoplasm. There is no luminal proteasome. So that sets the stage for the solution to the problem and the name for the phenomenon in general, which is referred to as retrotranslocation. And as a more general description of the overall process that we're talking about dealing with misfolded and no longer wanted proteins is the term ERAD, endoplasmic reticulum associated degradation. That phenomenon is shown on the diagram down here, beginning in the lumen with this thing, which we'll now call a misfolded or unwanted protein, has the glycosylation, the oligosaccharide trees, disulfide bonds, and there is BIP associated with it. It turns out that those components, while they are assisting the folding of some proteins, are also escorting unfolded proteins, misfolded proteins, unwanted proteins to the membrane protein in the lumen of the ER, in the ER membrane, that is going to lead to the export of that protein into the cytoplasm, from the lumen into the cytoplasm. So this complex actually is probably comprised of several different types of protein. It's not as simple a phenomenon as with the translocon, the Sec61 translocon. And the evidence for the things that are referred to in this diagram as the protein translocator complex is fairly weak. And there's some indication that Sec61 itself can participate in retrotranslocation in some cases, but clearly not in all of them. So exactly what this protein is not nearly as clear as is the case with translocon. But that something forms a pore through the membrane is a necessity that has just got to exist. What is agreed upon by pretty much everyone is what happens on the cytoplasmic surface, and that is that there is an E3 ubiquitin ligase sitting there either as part of the translocon or interacting very closely with it on the cytoplasmic surface, such that the misfolded or unwanted protein, as it emerges into the cytoplasm, picks up ubiquitin residues. And that process is also assisted by a AAA ATPase. Remember the hexameric ring structure that we've talked about several times, providing the energy and also unfolding the misfolded protein as it emerges into the cytoplasm. So there's one additional component that we've got to deal with, and that is that oligosaccharide that was introduced early on in the co-translational translocation process. There are cytoplasmic enzymes that remove those carbohydrates, and they're referred to collectively as N-glycanases. So once all of those activities have kicked in, then you end up with a very nice substrate for the proteasome, ubiquitin and partial unfolding triggered by the AAA ATPase. The cap can handle the rest, and the protein is destroyed. So that phenomenon is retrotranslocation, this part, and then ER-associated degradation is the overall process. There is an additional component to this phenomenon of ERAD that goes by the name unfolded protein response. And it's largely a gene regulation phenomenon, so we don't do those in this class. But I did want to mention it because what happens is that the presence of increased numbers of misfolded proteins in the lumen is actually signaled to the nucleus to synthesize more endoplasmic reticulum chaperones, such as the so-called lectins that bind to the carbohydrate, BIP, and disulfide isomerase that removes those disulfide bonds so that they can be retrotranslocated out. That's a very complicated process and quite bizarre that you have that kind of communication. But I did want to at least mention the term because it is a fairly important aspect of the stress response of eukaryotic cells in general, and it fits into the overall discussion of retrotranslocation.